All right, so welcome to the last session of our virtual tree fodder seminar. My name is Steve Gabriel with Wellspring Forest Farm, and really glad to have everyone join us live and watch the recording of this at a future date, but just to share this info and to talk about all things tree fodder. This event and the larger grant that our farm was supported with comes via Northeast SARE, and so I just want to give some acknowledgement that this work is based on funding from the National Institute of Food and Agriculture, U.S. Department of Agriculture, through the Northeast Sustainable Ag Research and Education Program under subaward number FNE 19930. We're happy to have Ashley Conway here. I'll let Ashley introduce herself and share about tree fodder research, and then we'll take some time to have some questions and some conversation. So let me pass this over.
All right.
Okay, you should be able to share.
Here we go. My screen shared here. Oh. This is the presentation version, isn't it? That you guys can see.
Yeah, I had this issue earlier too. Yeah, we can like see the.
All right. No, I can fix this.
Both slides.
Share this screen instead. All right. Did that fix it?
Yeah, that's great.
All right. Well, thanks, Steve, and thank you everyone for being here. Good afternoon, and I am happy to be here. And a little bit earlier, I got my time zones crossed, but hopefully what I can share with you all today is a good cherry on top to what has been, I expect, a really, really good session on tree fodder. I was asked to do sort of a research perspective on where tree fodder is and what we're looking at and some visions going forward into the future. So with that, as always, if you have questions, please speak up if you can, or pop them in the chat. I'll try to keep an eye on it as we go along. And keep in mind, if it doesn't happen as I'm talking, keep it in mind, and we'll bring it up in the question and answer session after.
Ashley, this is Mike coming out of the ether. I just wanted, because having been listening earlier, so one of the things you might cover also is how you would think about, doing a study to ensile some vegetation in case you wanted to carry it over the winter or something, because that has been brought up.
Thanks, Mike. That is an excellent idea. And in fact, I will get to that.
Okay.
I am going to start with sort of a broad perspective. So understanding that you did have some sessions earlier on the role of fodder in livestock production. Hopefully I won't spend too much time on this, but I want to get us all caught up to speed from a research and scientific perspective of why this distinction of what tree fodder is really important and why it matters. So, starting out, when we talk about tree fodder, it can be kind of a nebulous term. These are the definitions of forage and fodder. And as we can see, they're essentially the same if you go by just strictly the definition of like at a dictionary level, what forage and fodder is. From our perspective, and I say our as a research particularly revolving around animal nutrition and livestock production, from an applied perspective, the key difference between when we talk about forage and fodder, it may feel like semantics, but when we're talking about forage, we're really talking about grasses that are grazed in a pasture. And so we're looking at more cool season and warm season grasses. They can be perennials, they can be annuals, but the actual act of grazing and foraging for the forage is sort of in our mind creates this distinction of what a forage is. From an applied perspective, that difference between forage and fodder, we're really looking at woody temperate perennials, which is why we're here. So I hope that at this point is very clear and well understood. When we refer to forage and fodder, we are talking about different types of plants, and with that, the animal eating those different parts of the plants. You know, when we look at fodder, we're talking about primarily the leaf structures of these plants, which leaves really woody stems behind, and the animal's ability to browse and eat that fodder is an important part of this conversation as well. Something that I won't talk about here that you might hear in vernacular, if this hasn't been brought up already, fodder may be thrown around with sort of a new technology of hydroponic productions. So using quick growing cereal grains like rye in a hydroponic tray to grow really fast, succulent, lush little forages, and then feed it as a fodder source. Again, I think that's not really what we're talking about here today. We're really going to be focusing on the tree and woody perennial aspect of fodder. But again, we're looking at a supplemental feed stuff that's not foraged and grazed. So we're all on the same page with what this definition is, but when we look at the history, and with the history, what scientifically that looks like, fodder banks and stockpiling is really a long tradition of the history of feeding tree fodder. So this is not a new practice. Feeding of tree hay or tree fodder is really rooted in a lot of indigenous practices around the world. And most of the literature that you find on tree fodder occurs in other countries outside of the US, particularly in sub-Saharan African countries or in Southeast Asian countries. Nepal has a particularly robust tree fodder history and system of feeding that's integrated into their indigenous and traditional livestock keeping practices. And as I was doing more research for this particular topic, a really good example comes out of Nepal, where it's like built into their language. And Mike's here, so he can speak to this because I think he was involved in some of these papers. The indigenous practice and indigenous knowledge surrounding tree fodder and feeding tree fodder in Nepal is part of their language. And some papers were looking at how this correlated with the actual nutritive value. So the word, and I might not pronounce this correctly, so if anybody is listening to this recording or to this presentation and can correct me, please do so. Posilopan and Obanopan are the terminologies and the descriptors for these different species, like different types of feeding species that farmers in Nepal would use. And literally translated, Posilopan means high nutritive value, and Obanopan translates to dry and warm. So the Posilopan fodders were sought out and added to their diets to increase the milk production. It's thought to have an impact on butterfat content of milk and have a higher protein content. And the Obanopan is sort of a filler feed. So they would feed it to keep the animals dry and warm, understanding that it may not have as high a nutrient content, but it was sort of just to fill their bellies up when they were hungry. So this would be more common. You'd find more Obanopan species in the off season when things aren't quite as green and lush. And a couple of these studies are really looking at correlating these descriptors that the farmers in this study were looking at for the species, and then actually doing like proximate analysis and feed analysis to find out if this indigenous knowledge and understanding of how to feed these tree fodders correlated at all with the scientific nutritive value. So here's a list. This is from this paper from Thorne and others. We have a list of these tree fodder species that are common in that area of Nepal, a lot of ficus species, as you can see. And they talked to the producers, separated them out into having very Obano characteristics. So again, that sort of filler feed to keep the animals full and happy. And then the high nutrient content species down here, the Posilo species. And what they found is that there didn't seem to be much of a correlation between these various nutritive values in the Posilopan. There's quite a bit more variability there, but there was some correlation between this characteristic of Obanopan and the fiber content and the protein content of these ficus species. So there's a couple of things that we can draw from this study and the other studies that I looked at. But for me, I think the key points that I want to bring up here are that one, indigenous knowledge is indigenous science. And so there is value in understanding how traditional feeding systems work. And there's a lot of information that we can gather just from listening to other people and not necessarily listening to people like myself who are just strictly looking at one particular way of knowing, which is very lab-based. There's a lot of other factors that go into the value of a feedstock beyond just the nutritive value. And I think that that's what this study really pulls apart. So, and it kind of gets back to why we want to consider tree fodder as a whole. You know, there's more than just nutritional quality when it comes to feeding any feedstock really, but especially with tree fodder. We're looking at the value of including trees and temperate woody perennials as part of a system. And with that system offers a wider seasonal availability of feedstuffs. So this chart here, some of you may have seen a similar chart and many different options here. I picked this one from Florida, down in Florida, because it did have a lot of different species on it. But this throughout the year tracks the seasonal variability of different forages. And the common comparison you'll see is the difference between cool season and warm season grasses, and how you can sort of have some seasonal, spread out your grazing season throughout the year. But what I would like hopefully for people to pull out from this is this winter season here, we have stockpile forage. And stockpile grazing is a common practice here in the United States and in other parts of the world, but it sort of fits into this fodder conversation because we're talking about having nutritious feed sources for our animals that isn't necessarily, it doesn't have to be a forage, it can be these tree species at a time of year when there's not grazing available. And a lot of woody perennials, they aren't necessarily deciduous, or they have an extended season where they aren't prone to the same seasonality that green forages are. So you do have fresh forage and fodder available for them, for livestock to eat at times of year when you don't necessarily have grass grazing. So this picture here is a picture of some stockpile fescue, and you can see that it's definitely clearly winter time. There's not a lot of tree leaves on these particular trees, but we have grown some stockpile forage here for the animals to eat. So it's this concept of seasonal availability, climate resistance. And I use this term with the caveat that I don't necessarily mean climate change resilience, but resilience to seasonal droughts or flooding that may affect your forage availability. Forages can be pretty sensitive species, if I'm being really honest here. And I'm sure that many of you, this is not new information to you, but temperate tree species can be quite a bit more flexible when it comes to seasonal conditions and growing conditions and the stresses that are then applied due to weather, creating a wider, again, increasing the availability of feedstuffs for your animals. And then going back to, again, this knowledge that comes from utilization of alternative feed sources, this really fodder trees and fodder banks fit into these mixed purpose systems. So you have really multi-level and multifunctional opportunities for your livestock when you incorporate tree fodder or fodder banks or stockpiling. You just have a lot more flexibility, and adding those temperate crops, those temperate trees, improve the functionality of the system as a whole. You know, this is the stump speech about agroforestry in general. You have a multifunctional system where the trees are offering more than just fodder. They're also offering shade. They're offering allelopathic effects with the other trees around them. So if you're familiar with the term plant guilds, that's a pretty common term in permaculture world, where you have guilds of trees and grasses and shrubs that grow really well together and symbiotically exist. So, you know, thinking about the role that tree fodder can play in your animal system is really more than just nutritive value. Are there any questions at this point? I'm kind of cranking through this, so I want to make sure that everybody's caught up. Feel free to drop them in the chat at any time. So there's more than just nutritional quality, but of course, when we're looking at feeding animals, nutritional quality is pretty important, right? So these next few slides are, I'm pretty excited about this because I get to go back to sort of where my area of real training is, which is in ruminant nutrition, and why this is such a exciting area of study for me at the Center for Agroforestry. So again, if this is not new information to you, I apologize, bear with me. And if it is, also bear with me because I'm going to hit you with a lot here. So we are familiar with rumens in ruminants, and the ruminants have 4 chambered stomachs that act as a fermentation vat for plant species. So the niche of ruminants is that they are able to break down plant material that most mammals really can't. And the way that they do that is through this symbiotic relationship with microbes, anaerobic microbes, bacteria and protozoa and yeast and fungi that live in the rumen in this constant soup. And they are actually the entities that break down these compounds, these plants. And through this fermentation process, and this is a simplified schematic, you have various bacterial species that break down these plants into end products. And this is really the key output. The end products of fermentation are volatile fatty acids, primarily acetate, butyrate, and propionate. And then the animal can absorb these VFAs through the bloodstream and utilize that as a source of energy. So this whole idea that each ruminant has a billion little pets inside of their tummy that are doing really all the hard digestive work, and then they get to scrape off the top, and then in return, they provide a happy home for all of these really, really awesome microbes. To me, it's just really amazing. The cat's meow is my favorite part. And what really makes this cool, beyond just this, it's fun. We got to think about the structure of a plant. So as I said, mammals can't really break down the structures of a plant. When we eat plants and salads, it's called fiber, and most of it is indigestible. We have soluble and insoluble, but for the most part, if we eat a big salad or a nice broccoli laden dish, that fiber is going through our system and providing a lot of benefits, but we can't really break it down and utilize that for energy. Ruminants, on the other hand, because of this fermentation and symbiotic relationship, they are able to break down these plant structures and utilize that for energy. So to back up here, this is a picture, a microscopy picture of some plant cells. And I like to think of them as gushers. If you're familiar with the candy gushers, you have sort of this hard shell around the outside, and on the inside is all of these sugary goodies that are really rapidly fermented, highly nutritious. They're fast energy, and this is a good source of energy for the microbes, and they just go bonkers when they get to the inside of these cells. But to get to the inside, you got to get through the outside. And unlike gushers, where it's a nice chewy outside, it's really actually more of a fortress. So this picture over here sort of walks through what the bacteria have to break down and break apart to get to that gooey, yummy center with all of these super readily fermentable carbohydrates. And what it is the cell wall. And cell walls are this really intricate and highly bonded matrix. It's really a fortress, if you will, that also is comprised of sugars. If you get all the way down to the bottom, they're made up of glucose monomers. But the way that they are bonded together is a simple hydrogen bond that makes it stack on top of each other in this really strong matrix. And then that matrix is attached with more really strong bonds. And then this matrix here is the part that makes it undigestible for us. We can't break this apart. You need microbes to break these down to get at these glucose monomers. But because ruminants are wonderful little ruminants, they can actually break down these structures fairly readily through fermentation and use the microbes to not only get at the center parts of the cell, but also break apart this fortress, this matrix that's virtually indestructible for our intents and purposes. And that's the magic. So at this point, if this is clear to you, you may be wondering, why are you talking about this? Why does this matter when we're talking about tree fodder? For the most part, and these are general rules of thumb, when we look at the structure of the plant, we can make the general assumption that if there's more of this scaffolding, there's less goodies. So when I say scaffolding, I'm particularly referring to this external structure that includes cellulose, hemicellulose, and lignin. So it's scaffolding because this strong bond that creates this lignified structure, that's what makes plants stand up on their own and gives them the structure. It's the skeleton, if you will, of the plant. So if a plant has more scaffolding and more structure and more of these structural carbohydrates, then there's really going to be less space for all of the goodies in the center of the gusher, right? And we kind of know this intuitively. If we look at this lush young pasture here, we can intuitively know that there's probably going to be more nutrients for a grazing animal than this older dead pasture here, this range that's pretty scrubby. These plants are senescent, so it's past their growing season. A lot of their sugars have been moved down to the roots at this point to store, and what's left is really the skeleton of the plant. This is what allows it to remain, and these are all the structural carbohydrates. Now, a ruminant can still graze this and gain energy from this. It's not great, but again, intuitively, we know that they're going to do a lot better and gain a lot more weight on this lush green pasture compared to this dead, scrubby rangeland down here. So I also think that a really good way to tear this apart as far as plants, parts of the plant, is to use the corn plant as a really good example of how structures vary and how that affects the nutritional quality of the different plant parts. So I'm sure this is not a new picture. You're all familiar with what a corn plant looks like. And we know that if we feed the corn kernels themselves, it's a high energy feed. It's a concentrate feed. The corn kernel itself is almost 98% TDN. It's really high when processed in different ways. Of course, that's the caveat. But this is the fast candy bar of the ruminant world. The other vegetative parts of the plant, such as the stem and the leaf and the cob and the husk, these all vary, though, in structure and thus nutritive value. So intuitively, if we think about scaffolding equaling goodies and nutritive value, the leaf is going to be more digestible and have greater nutrients because it's less lignified than, say, for instance, the stem of the plant. And this really bears out if you do an analysis of different corn plant parts. This is from actually my PhD dissertation, and you can see that the husk and the leaf are the most, when you exclude corn, obviously. So we're talking about just the parts of the plant. Husk and the leaf are much more digestible than the cob and the stem. And a lot of that has to do with the structural carbohydrate content of those plant parts. So again, if you're wondering what this has to do with tree fodder, think about the nature of what we're feeding when we feed tree fodder, right? So this is some Glircidia, and here is some mulberry. When animals eat, they're taking big mouthfuls like this goat, and with some level of discernment, they're able to select their own diet, but oftentimes they'll get bits of this young stem in here. Maybe they're really bored and they munch a little bit up here to this highly lignified twig part. And even here is Quercitia, like that's a strong, oops, my bad. This is a strong structure of the plant. It has probably more lignin and more structural carbohydrates than this soft, lush leaf part. But a hungry steer that's coming in here and munching on this, there's probably munching all the way down here. So why this matters and why I'm providing this information to you is when we talk about tree fodder, woody perennial fodder, a lot of these species have very distinct plant structures and different plant parts. And because of the nature of of those different structures and how the plant is just physiologically, those will have variable nutrition. And it's much more distinct than when we're talking about like a forage species, where you have some stem that's closer to the ground versus the top of the leaf that's a lot more nutritive. The variability between these different parts of the plant is much greater when we're talking about tree fodder compared to traditional forages. And then how the animal eats it also makes a difference. So I want to provide you a little bit of information when you're interpreting your forage analysis, and I have forage in quotation marks. You can take samples of your tree fodder and send it into a lab to get it analyzed using traditional forage analyses. And then what you'll get back, if you haven't gotten one of these back before, you'll get back some information about what that sample is made up of and what it constitutes. You'll get a reading or a metric that's called dry matter. And what they've done is that they've measured essentially the amount of water in the sample through evaporation and difference. They've made a calculation that estimates the moisture of that plant. And water is a nutrient in the sense that we need it to survive, but it doesn't really provide any additional like protein or minerals or anything like that. But it is a factor in how much that animal can eat. So it will give us information on how concentrated the nutrients are in that plant and how much of that plant that the animal can eat. And if they're able to eat enough, will they get enough of those nutrients? I always like to use the analogy of Kool-Aid with dry matter. If it's a new concept to you, you buy a Kool-Aid packet and you have the concentrated dry matter of the Kool-Aid material. And you can mix up your Kool-Aid to be as dilute or not dilute as you want. So if you mix up one packet of Kool-Aid in a gallon of water, you're gonna have a very different drink than if you mix up one packet of Kool-Aid in 10 gallons of water, right? And if that one packet of Kool-Aid represents all of your daily nutrients that you require to meet your needs in one day, which of those two drinks are you going to want to drink? You're going to want to just drink a gallon and meet all of your needs, right? And so the amount of water and the amount of dry matter in a forage or in a fodder has a factor in how much you can actually eat to get the nutrients that you need. You might also see crude protein. You're probably going to see crude protein. And what this is precisely is that they've done an analysis where they measure the amount of nitrogen that you submitted in your sample. And through a calculation, they measure the amount of nitrogen through combustion. And then with a calculation to adjust, this value estimates the concentration of amino acids in the plant sample. And through that, you can sort of get an idea of how much protein is in that forage. Now, the caveat, why is it crude protein? Because it's an estimate of the amino acids. It doesn't necessarily tell us if those proteins are available for digestion, if they're bioavailable at all, or if they're even like intact. So the proteins, can denature, and that does change how digestible they are. So it's a crude estimate in every sense of the word. You may also see neutral detergent fiber and acid detergent fiber, NDF and ADF. And if you think back to that initial picture of the plant structure, NDF is a measurement that estimates the fraction of hemicellulose in the sample. So hemicellulose is the sugars that connect those really insoluble cellulose matrices. And the acid detergent fiber is a measurement of the cellulose in the sample. I can go back here just to help visualize. So we think about these lignin arms here in between the matrix and hemicellulose with the lignin that binds together the cellulose matrices, and all together it makes this really strong wall. So when ADF and NDF processes are run in the lab, it does its best to chemically break apart all of these different components and estimate what proportion of it is cellulose, what proportion of it is lignin, and what proportion of it is hemicellulose. Because depending on how lignified a structure is, this picture might look very different, right? So we can try to visualize these yellow blocks being much bigger, or the red strings being much bigger, or there being a lot more or less of the blue surrounding them. So it really varies between each different plant. You might also see in your forage analysis measurement of water-soluble carbohydrates, or WSC, or a variation thereof of that terminology. And this measures the fermentable sugars in the sample. So think about the inside of the gusher, right? We have the structural carbohydrates and we have the non-structural carbohydrates. And this is one way that labs will analyze that. So it can give you a sense of how big your plant gusher is, essentially. You might also see ether extract. I have only included it for your reference. It doesn't really give us an indication too much of nutritive value, in the sense it measures the lipids. It's not super common in forages to really matter. It's more common in co-products, but it's just so that you know, it measures the fats and plant waxes and of the plant. It does go into these calculations here. So I've included these two. These are the two most common calculations you might see on your forage report. TDN, the total digestible nutrients, and relative feed value, or relative forage quality. These are all calculations that are derived from a lot of different studies that have correlated through empirical studies what each of these measurable metrics, like water-soluble carbohydrates and NDF and ADF, with how well it's digested in an animal. So these are calculated values and they are estimates, and they are intended to give you an index, or an idea of what the quality is of your forage. But it's important to remember that it's not an actual measurement. So you have these numbers. How do you read them? Like, what do they actually mean as far as the value of your forage? So I found this really, really excellent chart from University of Georgia. So please, if you want to find more info, the University of Georgia Extension has an excellent website that it does include this page on interpreting your forage analysis. And I mostly just included it to give you an idea that these numbers that you'll get back, they indicate a range. And the range, if you fall within the range, then it's an average, but your sample may not. And so it's important to keep in mind that there's always a range when it comes to your feeds. And each type of feed has a particular range. So here we have neutral detergent fiber, or NDF. You'll see that small grains and a lot of the grasses are over here. They'll typically have NDF in the 50s to 60s range. But down here we have alfalfa that typically has very low NDF. And that's coupled with also very low ADF. So legumes, and alfalfa in particular, I wanted to point out, because they tend to sort of not fit with other traditional grass-based forages. And if you think about it, this is sort of a jumping off point for our tree fodder discussion, because if you think about the physiological structure of the alfalfa plant. You have small leaves that are highly digestible, very high in protein, and then they're supported by a more lignified stem. And when you cut alfalfa hay or you graze alfalfa hay, the animal is consuming both of those parts together in one mouthful. And so they're getting very, very densely nutritious leaves with less dense stems. So when you average those out, you end up seeing some results that sort of put alfalfa in different parts of the chart compared to other forages here. And finally, when you're thinking about it, I want to just add that the idea of a good forage and a bad forage, please banish the idea from your head, because the numbers are just numbers that can help inform you how best to include it into your routine. So think back to those farmers in Nepal that are feeding tree fodder, and understanding that it's not always a matter of a good quality forage or a poor quality forage, but it's a feed that has a function. So is it a highly nutritious feed that will provide additional energy when you need more milk, or you need to grow your animals a little bit faster for meat? Or is it a feed that will keep them full and warm in the winter and really not worry about improving like their performance beyond what they need at the time, if that makes sense? So why do we want to analyze our samples and our tree fodder? So this is explicitly for tree fodder. And it comes back to this discussion of, when I was thinking about how to present this particular topic, it's important to remember that tree fodder in itself is extremely diverse, and there's not one particular chart that I can compile for you that says, if you feed mulberry versus elderberry versus moringa versus Leucidia, like here are the values that they have been found to average in a lab, and here's how you would feed it. We don't have a lot of great information on that, at least in Western literature, that I can compile into a numerical database for you. And part of that is because there's so much variability, and this is really due to plant growth, and keeping in mind that the nutritive value and the metrics that we get from our plants when we send them in to be analyzed, it's affected by so many things, including growing conditions, the actual soil it's grown in, the time of year that you're sampling. So just within a particular species and location, there is considerable variability in what you might find for the nutritive value of that feed stuff. Also keeping in mind that tree fodder, we wouldn't be here if this was really a commonplace thing. I think there's growing interest, and that's fantastic, but up to this point in the US, at least, we have a tendency to really stick with commercial feed sources and more, I have domesticated and non-domesticated here in quotes, but the commonplace feed sources that we have, like fescue or orchard grass, these have smaller genetic variability because they've been bred for forage in a very specific purpose. And therefore, it's easier to collect a lot of different samples and get a smaller range of estimates. When we talk about tree fodder, we are talking about a huge genetic repository, a lot of heterogenetic expression, and therefore it's really hard to pinpoint. If you say, I want to know what the feed value is for X tree fodder, how do I feed it? How do I feed my willow? There really isn't a good feed table for tree fodder. Now, I have asterisks here before anybody corrects me. There is a European database, and it's here at, this is from the Netherlands. It's Voderbomen, I believe that's how you say it. And I have the link here. But I wanted to add that this is why we're having this conversation, because even though there is a database and they do have samples for a lot of different tree fodders, this is really data that's limited by geography. These are values that have been collected in fewer studies and done in a much smaller geographical location, so just in Europe. So you can use that as a jumping off point, but as it's important to remember that if you're looking up the value of willow, if it's been measured and evaluated in Europe, your numbers might be very different. So when you are collecting your samples, if you want to go out and see what you have for your tree fodder, make sure that you collect representative samples. So you want to go out, you collect from a lot of different areas on your farm, collect from a lot of different areas on the plant itself. And when you sample them, make sure you're cutting at a point where you are estimating your animals are actually eating from. So don't cut more twig than what they would eat. Don't only collect the leaves either, because they're probably getting some bites of that soft end of the stem as well. It is a non-exact practice, but you can try your best, and repetition creates representation. So I do have this website here. This Feedopedia is a great resource for you to check out. If you are interested in some type of tree fodder, this is a link to that tree fodder database in Europe. Keep in mind here, it does say that are relevant for European conditions. So again, anything you pull from this, take with a grain of salt. And if you have the opportunity and access, definitely want to make sure that you collect your own samples for evaluation. So I wanted to spend a little bit more time here talking about some work that we've been doing at the Center for Agroforestry, evaluating elderberry as a potential fodder source for tree fodder, and provide you with some preliminary information of what we've got so far. And one of the ideas for this is, we know that this is Sambucus canadensis. So if any of you are familiar, the European variety of elderberry is Sambucus nigra, and canadensis is the North American variety of elderberry. Based on the data that we have so far, the canadensis tends to have lower cyanogenic compounds, and the cyanogenic compounds themselves have the potential to be toxic to ruminants. Now, this is a whole exercise in what we're all here today to talk about tree fodder. So when I first arrived at the center, we had this plot available, and I was trying to think of a good study that involved feeding the forage or the fodder to ruminants. And I began looking to see how do I feed this? What is the nutritive value? And in fact, I could not find any good information about what the basic consensus on fiber and protein and these cyanogenic compounds were for elderberry, because it really hasn't been collected. And I think that's commonly what you're probably going to find if you branch off into a lot of tree fodder species. If there's information there, it's been singular collection events. So again, try to go back to that European repository, but keep in mind that those values probably aren't, when I was thinking about this study, they probably aren't very representative of these plants here sitting in New Franklin, Missouri. Particularly given that we know that cyanogenic compounds in canadensis are probably lower than in Sambucus nigra. So in reading more, a lot of the animal science literature said, do not feed elderberry at all because it has, the potential to be toxic to your animals. And you can't see it here in this picture, but right over here around the edge of this entire plot, there is a 12-foot deer fence to keep deer out from eating the elderberry. So somewhere in there is a disconnect between what animal nutrition literature is saying about the potential for elderberry as a fodder source, and with the reality of the situation. And deer can come in if they are allowed to and browse elderberries. So I decided to take 10 steps backwards here and back up and say, well, I'm not going to feed any of it yet. I'm just going to evaluate what is the nutritional value of elderberry at different times of year, and what are those cyanogenic compounds looking like as well? What's that profile? Is this plant going to be toxic if I feed it as a fodder source? So this was the premise of this particular study, and we've got we've just finished year 2 of data collection. So we're processing those samples now. And I wanted to have year 1 summarized for you all, but we're not quite there yet. But what I can tell you so far from what I've seen is that we have throughout the year quite a bit of variability in the crude protein amount and quite a bit of variability throughout the season of NDF and ADF and fiber, these structural carbohydrates. So we haven't done the cyanogenic analysis yet, but what I'm hoping to see is that what we end up having here is a very browsable forage throughout the growing season. But, you know, if you're growing elderberry for commercial production, you don't necessarily want animals in here grazing when these berries are producing, right? So how can this fit into an actual production system? So it goes beyond just the using it as a fodder source, as a tree fodder source, but in commercial production, it's common to prune or cut down the canes to about ground level every winter when these senesce and cut them back. So the concept of this second part was to evaluate the pruned material and ensile it and try ensiling it to see if we could create some elderberry silage. So if you'll notice here, we did this at the end of November last year. And what we found, that the goal was to have a few leaves at least. And this is mostly the twigs part of the plants. So moving forward, when we repeat this study, we're going to probably try to do this earlier in the season when there's actually leaves, like leafy biomass on these plants as well. But what was so interesting is that these look like dead twigs, but when we chopped it up, there's quite a bit of green material in here. And we packed these mini silos. We split these into 4 different categories, and we have 2 different packing densities. And for each packing density, one is inoculated with an ensiling inoculant and one is not. And really, when we started this, we just wanted to see, could we ensile this? Like, would this even work? And this is where my poor time management is unfortunately going to come kick us all in the rear end here, because I did have a couple tables on the initial results of the ensiling study that I wanted to share with you. And ultimately, what we did find was that these twigs ensiled, and it wasn't great silage, but it's silage, and it smells like silage, and it looks like silage, and there is definitely potential to even in the late season incorporate elderberry silage into this production system. Whether animals will eat it and what the digestibility of this is definitely still a question that we have, but we have some really cool numbers so far, and lots of potential for tree fodder in the future to incorporate as a feed source for silage. So again, I did have a few more slides that I wanted to share, but I didn't quite get around to them with my my little miscommunication on my schedule, and I apologize. But at this point, what Steve and I talked about is we really wanted to make this a Q&A, and I have a lot more information that I can share with you. So at this time, I will stop sharing my screen now and pass it back to Steve and hopefully answer some questions for you all.